Yes, Komal, come on. You wish to ask something? Can you please give a standard mode option? Like, what standard mode option? I can't see standard mode option out over here. Like, what standard mode option are you talking about? Are you able to see my screen, guys? Are you able to see my screen? Like, whatever I'm presenting. Okay, okay. Take it. One moment. One moment, guys. Don't worry. One moment. Hmm. Now is it fine, guys? Okay. Okay, so guys, we last class, okay, so we had done until like 200, like really properly, and then after that we just hurried a little up. Okay, so I want to finish those parts off so that you don't have any problem. So guys, I want you to solve this 203rd one. Okay, and guys, let me know what is the answer for this 203rd one. Yes, yes, guys, what do you think, guys? Quickly. Okay. So, guys, what do you think is going to be the answer? So, you here. Okay, let's try to see what is going to happen, guys. So, we are going to have here, we are going to have XH. We are going to have XH out over here. So, this is going to be XH, XH. Okay. Then, mm, this is also going to be the same thing XH, XH. Okay. This one. What is this going to be, guys? This one is going to be XH, Y. And here, this is also going to be XH, Y. Right, so guys, let's try to see what is the right option now. Yes, guys, what do you think is the right option? Quickly. No, someone is saying answer is D. Wrong. Answer is not D, guys. Answer is B. Check the options, guys. Check the options properly. The option is B. Oh my god, someone is in the waiting room, I guess. No, I've already taken everybody in. Okay. Okay, guys, next question is 205. Okay, so we have to pick up like the correct statement. Okay, so guys, first tell me. Okay, pick out the correct statement. Hemophilia is a sex linked recessive disease. Is it true or is it false, guys? Pick out the correct statement. Is it true or is it false? Yes. Give me T or F, guys. T or F. T or F, guys. T or F. Yes, very nice. Okay, someone is saying false, some of them are saying true, but yes, this is true, guys. This is true because hemophilia is a sex linked recessive disease because we had said this is going to be XH. Okay, so XH, this is your defective. Okay, so this is recessive. Okay, fine. Down syndrome is due to aneuploidy. What do you think, guys? Yes, this is due to aneuploidy only. What is aneuploidy, guys? Aneuploidy is nothing but when you have one or more chromosomes, okay, or one or less chromosomes, okay. So it is going to be aneuploidy. So yes, it is true. Down syndrome, what is it going to be, guys? It is 21 trisomy, meaning the 21st chromosome is going to be 3 times rather than 2 times. Okay, so yes, this is going to be aneuploidy. Phenylketonuria is an autosomal recessive gene disorder. What do you think, guys? Yes, very nice. This is going to be true. Once again, this is directly from the text, guys. Okay, if you remember, again, if you keep it in mind, guys, these few examples, you need to know them by heart. Okay, and finally, sickle cell anemia is X-linked recessive gene disorder. Is it true, guys? True or false? True or false, guys? Yes, this is going to be completely false. Why? Because this is your autosomal recessive disorder. Okay, so this is wrong. So what is the answer, guys? 1, 2, and 3, they are going to be right. Okay, so yes, 1, 2, 3 are right. Okay, next question, guys, is yeah. Okay, so to 209. Okay, so let's try to solve this thing, guys. Down syndrome, guys. Down syndrome. Which of these is correctly matched? Which of them are correctly matched, guys? Let's try to see this thing. So Down syndrome trisomy 21, is this right? True or false? True or false, guys? T or F? T or F? Quickly, quickly. Yes, this is true. Okay, Klinefelter syndrome 45 and XO, is this right, guys? Is this right? No, this is wrong, guys. Rather, what it should be, it should have been 47 and XXY. Right, this should have been. And Turner syndrome, is this right? No, once again, no, because Turner syndrome should have been 45 and XO. So once again, guys, only one is the right answer. Okay, 210, guys. This one also very important. Yes. Okay, so the gene for hemophilia is located on the X chromosome, hence it is normally impossible for. Yes, guys, tell me, what do you think? No, 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 no. Someone has given me the wrong answer. Guys, tell me, what do you think? Carrier mother to pass the gene to her son, hemophilic father to pass the gene to his daughter, hemophilic father to pass the gene to his son, and carrier mother to pass the gene to her daughter. Yes, she has given me the right answer. Ashita has also given me the right answer. Waiting for the others, guys. Waiting for the others. Yes, Devank is right. Himanshu is right. Only a few are right. Okay, guys, what is the answer? Yes, the answer is going to be C. Hemophilic father to pass the gene to his son. Because see, guys, what is this? This is your XH, right? XH. Why? But now, when the male is going to give rise to a son, what is going to happen? So let's say he marries a normal female. From here, it is going to be X. From here, he is going to give Y chromosome to the male. So you see, when you are going to have a male child being born, who is contributing the Y? The Y comes from your father. And that is why hemophilic father can never give the gene to his son. Hemophilic father will always give the gene to his daughter. And that is why, what did we say, guys? We are going to see that zigzag inheritance or your crisscross inheritance. Okay. Now, guys, next question is 212. Okay. Sickle cell anemia. Guys, what do you think? Okay, I have told you, we talked about this a little earlier as well. So we had said, guys, sickle cell anemia, we are going to see that, oh, we are going to have, you know, the 3, 4, 5, 6. Okay, 7. Let's imagine. So we have your entire protein like this. Okay, so we have your protein now. General normal protein, guys, what is going to happen at the sixth position? At the sixth position, what do we generally have in your hemoglobin? It is glutamic acid. It is glutamic acid. But now, guys, what happens is when we talk about sickle cell anemia, instead of glutamic acid on the sixth position, now there is going to be a small change in the amino acid. Okay, so what is going to happen here, guys? Now, instead of having this glutamic acid at the sixth position, guys, we are going to see valine. And how does this thing happen, guys? This is going to happen because of only one single base pair change, guys. It is going to be single base pair change. So, can you tell me what is this going to be? Yes. Yes. Some of them they are giving me the right answer. Some of them they are still giving me the wrong answer, guys. But yes, you are going to be right. Most of them they have given me the right. The answer is substitution. So instead of one, we have something else. So that is nothing but substitution. Okay, everybody all right? Okay. Sickle cell anemia, a hereditary disease, was first described by. Now, this was not there in our syllabus as such, but then can anybody guess what can be the right answer, guys? Anybody? Can anyone guess? A, B, C, D. Yes. Yes, yes, yes. Now, the answer is A. It was James Herrick. Okay. Down syndrome is also caused by substitution. No, 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 no. Down syndrome is your chromosomal disorder. It is caused by non-disjunction of your chromosomes. Guys, this question, okay, the 218 question, this has been asked again and again, again and again, again and again, in multiple multiple exams. Okay, but once again, what I want is, guys, see, while we were doing this thing, I had told you. So tell me. Yes. Very nice, guys. Very nice. Answer is malaria. Okay, it was actually a mutation to get rid of malaria. But then it got rid of malaria. But in fact, what happened? The entire RBCs were only non-functional. Okay, my question is, guys, 219. This was the last thing that we had, like, solved, like, really in depth. Has everyone understood this 219th question? Quickly chat and answer, guys. Yes or no? Okay, 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 okay. Could you solve it again? Okay, no problem. Okay. So once again, guys, in this thing, you just have to, go with the flow. Like, whatever the person is saying out over here, you just need to jot it down. Okay. So a man whose father was colorblind, okay, marries a woman who had a colorblind mother. Now, guys, this statement is not like, a man whose father was colorblind, meaning the father is going to marry a woman. It is the man who's marrying a woman. Okay, so understand this thing. One year there was a kid, who came and you told me, sir, that man is not marrying, the man's father was marrying. And I said, no, 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 it is not that way. There should have been some proper, like, this should have been put in a proper thing. But no, it is not that way. Okay, so guys, keep this thing in mind. A man whose father was colorblind. So let's say we are saying this man, this is XY. his father was colorblind. So if he was colorblind, he is going to be XCY, right? So if this guy is XCY, guys, is he going to receive the defective chromosome, the defective gene? What do you think, guys? Y or N? Y or N? Yes, absolutely. It is no. He is not going to receive the defective gene. Okay. So basically, he's like this. Okay. A man whose father was colorblind marries a woman who had a colorblind mother and a normal father. So let's say she is this way. Okay. And she had a colorblind mother. So it was XCXC and a normal father. So normal father is XY. So what is she going to be, guys? She is going to be XCX or XXC, because from here you're going to get this thing, and from here you're going to get X, and from here you are going to get Y in your name. So now, what percentage of this couple will be color blind? So now let's try to see this thing, guys. Okay, so we are going to have X, we are going to have Y, we are going to have X, and we are going to have XC. So once again, guys, we are going to have XX, XCX, XY, and XCY. Now, guys, your tendency now in this situation, okay, guys, your tendency would be to find out, oh, how many of them are colorblind? Oh, okay, okay, this one is colorblind. Rest all the three they are going to be normal. What is the percentage? You will say, oh, percentage is 25. Yeah, kachak. You will mark 25. Wrong. Why wrong, guys? Because what are they asking? They are asking what is the percentage of male child of this couple will be colorblind. Now, how many are male child, guys? We have two male childs out over here. Okay, one is normal, one is colorblind. So, what is the ratio, guy? The ratio is going to be 50-50. Okay, and that is why, yes, 50% are going to be color blind, 50% are going to be normal. Okay, has everybody understood now, guys? Yeah. Okay. Okay. This is also im- important name, but this is asked sometimes. 223. Yes. Okay. Which of the following set of syndromes show the chromosomes in their genetic makeup? Show show 47. Okay, this is 47. Sorry. Show 47 chromosomes in their makeup, guys. Okay, so we have guys, first one we are going to have Down syndrome. What is Down syndrome? Yes, this is going to be trisomy. That is 21 trisomy. Okay, then guys, we have something that is called as your Patau syndrome, and we have your Edwards syndrome. Now, what happens, guys, when we talk about this Edward syndrome? Okay, Edward syndrome is, first of all, like, the babies, they are going to remain small. They are going to have long fingers, like, a little different. That is going to be because of your 18 trisomy. Okay, so this is going to be because of 18 trisomy. 18 trisomy. One minute. Okay, this is going to be 18 trisomy. And guys, then after that, we are going to have your Patau syndrome. Okay, now Patau syndrome, guys, you are going to see that in Patau syndrome there is a cleft palate. Okay, Cleft palate meaning you are going to see that there is a complete mark, okay, onto the this thing, onto the no the jaw kind of a thing that is going to happen. And let me just show you the picture. I'm sure you guys must have seen this thing. uh, 1 minute, I'm just trying to locate a picture. Guys, see this. Have you ever seen, like, those who have like cut in their lips or cut in the upper jaw? Okay, that is nothing but this Patau syndrome, guys. Okay, so this is going to be because of your 13 trisomy. Okay, or I'll show you this. There is one more image which is like the proper image. See this one. You must have seen people with this kind of thing. This is called as cleft palate. Okay, and this is your Patau syndrome. Okay, this is going to be because of your chromosome number 13 trisomy. So actual answer, guys, is your A, that is your Down syndrome, Patau syndrome, and Edwards syndrome. Okay, just keep this thing in mind. Okay, it was asked, in one of the competitive exams. Okay, then okay. So guys, uh, 224. This one. Okay, genetic disorder hemophilia is characterized by excessive loss of blood. Which of the following statement is not true in relation to this disease, guys? What do you think? Okay, it is an autosomal recessive disorder. Is this true? T or F, guys? T or F? This is going to be false. So this is the first one itself is false. It is an X-linked disease. Yes, this is right. Any of the factor 8 or 9 may be absent. This is also right. It is a lethal disease. Yes, this is also right. Okay, someone is asking, sir, what is Edward syndrome? Edward syndrome is like, when generally seen in your females. Okay, and once again, like I said, this is going to be 18 trisomy. What you're going to see is like, if, you are going to have a little bit of your cleft onto your mouth, but along with that, you are going to have like, long fingers, clenched fist, guys. The ears are going to be not proper, and the eyes, they are going to be small. Plus, you're going to see that the head is comparatively small. That is what is going to happen. So if you want, let me just quickly show you a picture, a representative picture, guys. Okay, so see, this is the picture for your Edward syndrome. Okay, it looks somewhat more or like your this thing only, Patau syndrome, but here, see, the eyes are also small. The head is in, in total this thing, the head is small. The hands, they are going to be like clenched. Okay, the fist is like clenched fist. So let me just show it out over here. See here. Okay, so the fist is like, locked up kind of a thing. That is what is going to happen. Okay, and as you grow, guys, so something like this, you will be something of this sort. This is Edward syndrome. Okay, sure. Okay. The next one, guys. Next one is your glucose 6 phosphate deficiency. Okay. This thing is an inheritance, is an example of dash inheritance. So, guys, once again, you will have to guess this thing. We haven't done this thing, that is not there for you. But then, what do you think it is going to be? Most of the diseases, they are going to be recessive in nature. And I'll tell you that this is going to be your X linked recessive in nature. Okay. So, X linked recessive. Okay, this 227 we have already talked about this thing. But then last time also. So, person affected with phenylketonuria lacks an enzyme that converts the amino acid phenylalanine into dash. What do you think, guys? Yes, very nice, very nice. He has given me the right answer instantaneously. Himanshu is also right. Yes, it is going to be your tyrosine. Can anyone quickly give me what was that enzyme that we are talking about? It was for phenylketonuria. What was it? Phenylalanine. Quickly, 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 quickly. Come on, guys. It was. Come on, someone. At least one of you give me, guys. Quickly. Phenylalanine, guys, hydroxylase. Okay, so we had used that one out over there. Okay, so deficiency of phenylalanine hydroxylase is going to cause your phenylketonuria. Okay, this 229, guys. Okay, sickle cell anemia is caused due to the substitution of. Yes. Just, guys, quickly, what do you think? What should be the answer? Quickly, quickly, quickly. See, guys, we have said in your hemoglobin there are going to be two chains. We have the alpha chain and we have the beta chain. So here, where is the effect, guys? The effect is seen on the beta chain. So what is it? Okay, is due to the substitution of glutamic acid at the sixth position of beta globin by valine. Okay. Yes. Okay, guys. Now, 232, guys. Solve this one, guys, quickly. If both parents are carriers for thalassemia, which is an autosomal recessive disorder, what are the chances of pregnancy resulting in an affected child? So, guys, we have thalassemia. So, we have HBT. Okay, so let's imagine HBT, and the other one is also a carrier. So, HBT. Okay, so let's figure this thing out. So, guys, what are the chances of them resulting into? So, guys, this is nothing but your a typical type of your this thing selfing of your F1 generation that we had done, guys. So what is it going to be? So ultimately we are going to have HB. Ultimately we are going to be HBT, HBT, and lastly is HBT. Yes, so guys, the chances is nothing but it is going to be 25%. Okay, so see here, only this one is going to be the sufferer. Rest 75% they are going to be normal. Okay. Okay, 235, guys. Which of the following is an X-linked recessive trait with locus XQ28 and related with factor 8? Yes. So you will have to think about this thing, guys. Okay. First, what is this uh, XQ? Okay. First, I want you to guys read this thing. Then I will tell you what are these two arms of the chromosome. Okay. So, guys, what happens is whenever we talk about any chromosome, okay, so you are going to see that, oh, the chromosome, we have shown one single chromosome, something like this, guys. And let's say we are showing, okay, now the centromere is out over here. So, there are two arms, one is short arm and one is long arm. So, what we will see is this short arm of the chromosome, this is called as the P arm, and this long arm, this is called as the Q arm. So short arm and long arm. Okay, so just keep this thing in mind, guys. They will ask you, P is short. Arey, what is happening? Okay, P is short and Q is long. Okay, so what are they saying out over here is the chromosome is going to be X chromosome, and on the Q we are going to have somewhere on the 20th position, somewhere out over here. Now, guys, this is generally for your hemophilia A. You will have to remember this thing. Okay, so just keep this thing in mind. Always remember P arm is the short arm, Q arm is the long arm. Okay. Okay, next choose a false statement with reference to sickle cell anemia. Yes. So, guys, which do you think is going to be false? Yes, Devang has given me the right answer. Yes, others, quickly, quickly, quickly. Uh, yes, guys. Anyone? Chalo, let's try to see. First of all, in your sickle cell anemia, okay, guys, they have a genotype HBS. Is this right, guys? Is this right? HBS. Is this the genotype? What do you think, guys? Quickly, quickly. Yes or no? Yes or no? True or false, guys? Quickly, quickly. Hello, hello, guys. Can you hear me? Hello. There is no response from your end, guys. Okay, so what do you think? Is this right? Is the genotype going to be this? Or what did we say, guys? We had said it is going to be HBS. Why? Because this was a lethal gene. Okay, so this is a little dicey only. Okay, second thing, they have a genotype HBA. Okay, is this right? This is to 101% not right only. So HBA, no, this is not possible. Okay, have substitution of glutamic acid to valine. Yes, this is right. Have sickle ship RBC. This is also right. Now, guys, you have to select one of the two. Who do you think is going to be wrong? Who is going to be completely wrong? Let me put it that way. Yes, yes, absolutely, guys. So in this situation, guys, still they have a genotype HBS. You will see something out over there, but then they have a genotype HBA. No, this is completely wrong. So you will go with the B option. Okay. Chalo. Okay, guys, 239 represented below is the inheritance pattern of a certain type of trait in humans. Which one of the following conditions could be example pattern. Okay, male to female, female to male, guys. Quickly, very simple, guys, very simple. Where no 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 guys, see, male to female, female to male, guys. Only Sneha Singh and Himanshu is right. Okay, so what is this? This is nothing but your X-linked, and in X-linked, who is X-linked out over here, guys? It is going to be hemophilia. No, no, no. It is going to be hemophilia, guys. Okay, guys, next 240. A normal vision man whose father was colorblind. Okay, so once again, guys, I want you to solve this thing, please. Okay, everyone. Okay, a normal vision man. Okay, so let's say we are saying it this way. A normal vision man, his father was colorblind. Okay, so his father was colorblind. So is he going to get the colorblind gene? No, because father is going to give him only this thing. Okay, why? Okay, he marries a woman. Okay, he marries a woman whose father was also colorblind. Her father was also colorblind. So, guys, if her father is colorblind, then what is she going to be? She is going to be XXC, right? They have their first child as a daughter, right? Now, what are the chances that this child would be colorblind? Okay, chalo. So once again, so guys, we are going to have X, Y, we are going to have X, X, C. Okay, once again, we are going to have, see guys, X, X, we are going to have XC. Now, here things are going to end out over here only because we are not concerned with the male child. But still, if you want, I will write down XY and XCY. But now, what are they asking, guys? Girl child. What do you think? What are the chances that this girl would be color blind? What do you think would the chances be? Yes, it would be zero because there is no chance. Okay, so there is no chance because see, she'll be completely normal or she is going to be a carrier. But no matter what, even carrier is also normal only, right? So that way, so there is 0% chance. Okay, guys, this is also important. 242, the gene for diabetes mellitus. What do you think, guys? Okay, this is a little exception. Okay, this is exception, guys. Okay, but just think about this. Just think about this for a moment, guys. Take a little while. Okay, now see, guys, what I have told you is, see, most of the diseases, okay, most of the diseases, they are going to be recessive in nature. But guys, I right now told you this is going to be a little exception. This is going to be autosomal dominant. Okay, so guys, please keep this thing in mind. Okay, so this is very, very, very important. This is asked in your exams. Okay. Okay, next is 245, guys. We'll solve this one. If father shows normal genotype and mother shows carrier trait for hemophilia, then guys, think what will happen. Father is normal, female is carrier for hemophilia. So guys, just see what is going to happen. Okay, guys, just see what is going to be the right answer. All the female offsprings will be normal. Is this right? all the female offsprings will be normal. Actually, one is going to be a carrier, another one is going to be completely normal. Okay, but actually it is going to be a little right. Okay, all the female offsprings will be carriers. No. So this is also no. This is also no. A male offspring has 50% chance of active disease. See, male, this is diseased. This is going to have hemophilia. This is normal. So yes, this is right. And a female offspring has probability of 50% of active chance. No, there is 0%. Okay, guys, 247. Let's just see it once. What is this going to be? 247, guys, quickly. Yes, very nice. It is going to be XO. Okay, so it is going to be 44 plus XO. Okay, next question. Okay, next question. The gene of sickle cell anemia is inherited by. Guys, what do you think? A, B, C, D. Uh huh. Is inherited by. Guys, blood cells, bone cells, sex chromosomes, autosome. No, 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 guys. Sickle cell anemia, once again, is it going to be sex chromosome? No, it is an autosomal disorder. So it is going to be inherited by the autosomes. Okay, next one, guys. XXY. What is that going to be? Quickly. ABCD. Yes. It is going to be your C, that is Klinefelter syndrome. Right. Okay, guys, next one. Which of the following is not a Mendelian disorder? Hemophilia, color blindness, Turner syndrome, cystic fibrosis, and thalassemia. So, what do you guys think? No, 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 no. See, when we talk about your Mendelian disorders, what are these Mendelian disorders? The chromosome number remains the same, but there are going to be defects onto the genes or the alleles. Okay, and non-Mendelian are the ones where the chromosomes are only defective. So, what is it? Yes. Okay, chalo. 251 is also very important, guys. Due to non-disjunction, okay, due to non-disjunction of chromosomes during spermatogenesis, sperms carry both the sex chromosome 22A plus XY, and some of the sperms do not carry any chromosome 22A plus O. If these sperms fertilize normal eggs, okay, what types of genetic disorders appear amongst the following offsprings? So, guys, once again, what are they saying is like, we are going to have 22 plus xy, and other one is going to be 22 plus 0. Okay, these are going to be your sperms. Okay, and if this is going to meet with your 22 plus x ovum, then ultimately what are we going to get? We are going to get 22 plus 22, that is going to be 44 plus xxy. Once again, what is this? This is Klinefelter syndrome. And if the other one is going to meet, then it is going to be 44 plus xo. What is this? This is going to be Turner syndrome. So either your Turner syndrome and your Klinefelter syndrome. Now, guys, there is this thing which is written out over here, Cry-Do Chat syndrome. Okay, this is very important because Cry-Do Chat. What happens is here, the baby is going to cry like a small cat. Okay, and she is like going to make like weird noises, like just like a cat. So that is your Cry-Do Chat, guys. Okay, but now the question arises: why is this thing going to happen? Cry-Do Chat. Okay, the reason is, uh, you know We have chromosome number 5. Okay, so let's say we have chromosome number 5. I told you that, see, there are going to be two arms, your P and Q, right? P arm was the short arm and Q arm was the long arm. Let's say this is normal chromosome number 5. Now, what happens in Cry-do-Chat syndrome is the same chromosome number 5. Now, it is going to have a missing. Okay, it is going to have a missing P arm. Okay, so this chromosome number 5, now you are going to see that, oh, it is not going to have that P arm only. So you are going to see that, oh, the centromere is out over here and then the Q arm is out over here. This, and this is missing the short P arm. P arm of chromosome number 5. And that is nothing but cry to chat syndrome, where the babies, they are going to cry like a cat. That way. That's it. That is what we need to know, guys. Okay, and the rest of them, they were like really simple, so nothing to worry from here. Now, just a moment, okay, guys. I just need to check one of my books if I have something more on the pedigree chart, the pedigree analysis. Okay, so let me just give me a moment. I will just check if I have a book. Sir, if a question comes on a super male and female type, what are the parameters to solve? Now, generally, they won't ask you super male or a super female, because super male is actually, XXXY, then super female is going to be triple X. So they won't generally ask you on that. Okay, let me just, guys, quickly just see. Just a moment. I'm just going through that book. Let me just locate it. ओके गाइस, अरे ये कहाँ हो गया? There are not many, but I'll just show you one. Okay, guys, try to solve this thing. Yes. What do you think, guys? It is going to be very, very simple, guys. Very, very simple. Yes. Himanshu has given me the right answer instantaneously. Guys, others kindly answer quickly. Others kindly answer. Okay, and those who are done, I want you to solve this one as well. And there is one more. Okay, so guys, the first one. Okay, come on, guys. The first one. What is this? This is nothing but autosomal recessive disorder. How come, guys? See, look at this center part, guys. This is the biggest clue. See, what did I tell you? What is autosomal recessive? It is always going to skip generations. Okay, so see here, in the parent it is going to be present, here it is not present, once again coming back into your children. So what is this? This is your recessive. First of all, it is going to be recessive, right? So 99% we have already got this thing that this is going to be your recessive thing. Okay, but now let's imagine, uh, we are talking about, but now it is it going to be X linked or Y linked? How is it going to be? So Y linked, see, had it been Y linked, then what? The male is affected, the male is affected, the male is affected. But no, it is not that way. Here the male is affected, here the female is affected. So no, this is not a possibility. Okay, is it going to be X linked? So here, if male is affected, is the female going to be affected? No. And if the male is affected, once again, is the male is going to be affected? No, right? Generally that is not a possibility. So if this is not even your X-linked disease, so X-linked is not there, Y-linked is not there, what about your this thing, the autosomal dominant? But had it been autosomal dominant, guys, it would have been present in almost all the generations, but it is not so. So yes, then the next best option is autosomal recessive. Okay, yes. So, guys, this one. Study the pedigree chart of a certain family given below and select the correct conclusion which can be drawn for the character. What do you think, guys? Once again, this is very simple. Very simple, guys. Ashita is once again giving me the right answer. Yes, Ashita is right. Rest everybody is wrong. Guys, think over this. Think over this. What is this, guys? You are going to see that here. Okay. See, you are going to see that this female, she is not, this thing, but all her progeny, almost everybody is like, being affected. So what does this tell you? One is not affected and the rest others are affected. So basically this is also heterozygous. This is also heterozygous for the character, and she is not suffering, so she is going to be like this thing. She is also going to be this thing. So, you know, w- there are a few things that can happen. The female parent is heterozygous. Okay. But now, if the female parent would have been heterozygous, guys, even this male would have got the disease, something of that sort, if she was heterozygous, right? So, the male is not suffering out over here. So there seems to be some issue with this. Okay, so female parent is not heterozygous. The parents could not have had a normal daughter for this character. No, they could have got. The study under the control, could not be color blindness. Now, what do you think, guys? Yes. See, is this going to be color blindness? No, because from the male it is going to be given to the female, but are females going to be sufferers? No. Here you're going to see that, oh, this is also going to give it to the male. So I can definitely say, see, others I don't know. Other options I don't know, but here I can definitely say that the trait under the study could not be color blindness. बाकी का मुझे मालूम नहीं. Okay, so it is going to be that way. So only few of them had messaged me C. So whose ever name I had taken, like, very good. Okay, you analyze this thing properly. Okay, this is actually a trick question, guys. Okay, this was a trick question. Okay, and the last one, guys. Once again, see this thing. Once again, what do you think? Yes, 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 yes. Sia has given me the right answer. Yes. Swara has raised hand. Okay, sorry. 1 minute. Huh? Swara, yes. किधर गया? अरे, Swara, do you wish to ask anything? Okay, okay. No problem. No problem. No problem. Guys, what is this? This is once again your autosomal recessive. See, once again, who is the clue? Right in the first generation out of a year, parents are not sufferers, but suddenly the child is a sufferer. What is this? This tells you that yes, this is going to be recessive. Okay, both the parents are heterozygous and this is a recessive character. So once again, see guys, here parents are not sufferers, but child is a sufferer. So heterozygous. Okay, so yes, this is going to be heterozygous and recessive. So yes, this is going to be your inheritance, that is your autosomal recessive trait, just like your phenylketonuria. Okay, everybody all right with this? Fine. Okay, sure. So, guys, this was the last thing that I had to talk about. Like, last class, we had to rush this thing up. So I thought that we'll just take a little while and go through this once again. Okay. Anyways, chalo. So, guys, once again, what I'll do is I'll give you just a small 5-minute break. Okay, and then we will start off with a new chapter. Very very simple chapter: microbes and human welfare. It's a very nice, simple, fun chapter. Okay, so 5 minutes break, guys. Anybody wants to have go to the loo, have some water, quickly just have it and come. Okay, chalo. So guys, let's start once again. I hope everybody is back. Okay, so guys, the next chapter that we're going to start off. Okay, so that is going to be microbes in human welfare. Okay, so at this moment now this thing the slides, the PPT slides now, they are not perfectly ready, so I'll start off with this thing, and as we move ahead now, I'll introduce those slides in between. Okay, so first of all, guys, microbes in human welfare. Now, where is this chapter? If you look into your textbook, guys, you won't find this chapter in the table or the table of contents. Okay, so basically, you will have to look out for this chapter called as enhancement in food production. That enhancement in food production, guys, it is comprising of three different chapters. One, it is enhancement in food production. Second, it is going to be animal husbandry, and third is going to be microbes in human welfare. Okay, so guys, there you are going to get it. Okay, in your HSC textbooks, microbes in human welfare. Okay, so guys, the first thing that we need to know is whenever we use the term microbes, or whenever we use the term microorganisms, okay, so microbes or microorganism, who are we really talking about? 1 minute. In NCRT, it is microbes in human welfare or human organization. It is something of that sort. Okay, so guys, always remember whenever you use the term microbes or microorganisms, guys, who are we really talking about? We are talking about three kingdom out over here. So basically, we are talking about your Monera. That is nothing but it is going to include all your bacteria. Second, we are talking about your Protista. Okay, Protista is nothing but like your amoeba, your paramecium, etc. Okay, amoeba, paramecium, euglena, etc. And the third one we are talking about is your fungi. Okay, so it can be your yeast, it can be your aspergillus, it can be your rhizopus, penicillium, okay, anything, mucor, etc., etc., etc. Okay, so guys, generally when we talk about your microorganisms, they are going to be referring to these three kingdom. Okay, so keep this thing in mind. But now, guys, when we study these microorganisms, so what is their study called? So if you see this branch of branch of your biology, which studies microorganism, that branch is nothing but it is known as microbiology, the branch of science that deals with the study of microorganisms, that is microbiology. But now, guys, when we talk about microbiology can be of different types, guys. Okay, it can be of different types. I'm just giving you a few ideas. Okay, so the first one, it is going to be your normal microbiology, that is general microbiology. So where we are talking about, all the the life processes, the different kinds of your bacteria, okay, their properties, okay, all that thing, that is going to be your general microbiology. Okay, then let's say we go a little bit more into this thing, like the diseases and the disorder, then we call this thing as medical microbiology. Okay, fine. Then after that, guys, if by any chance, okay, if by any chance you are studying, using your microorganisms for producing huge amounts of your different kinds of products for mankind, then it is called as your industrial microbiology. Okay, and finally, if you're using it in the preparation of food, so then that is called as food microbiology, like that. Okay, and so on and so forth. So there are going to be many fields out over there, but then these are some of the basic fields that we have talked about. Okay, but now, guys, the first thing, okay, we are going to see, like, how are we going to use this microorganism? Where are they going to be used? Like, what happens? So the first thing is we are going to talk about the use of this thing, okay, the use of your microbes in your food production, and let's try to see how we will use them in producing food. Okay, so let's say this is number one. Okay, microbes in food preparation, or we can also say that this is going to be household microbiology, because it is going to take place in each and everybody's kitchen. That is why food preparation, or this is going to be household microbiology. Okay, so now guys, when we talk about this, what are we talking about? See guys, we are talking about like, oh, we are going to make use of these microorganisms to prepare our food. But how are we going to do that, and what really happens? So guys, the most common example that we incorporate in our kitchen is nothing but fermentation. Okay, and guys, what is this fermentation? Fermentation is nothing but this is going to be your anaerobic respiration. And guys, where are we going to bring about this anaerobic respiration, guys? We are generally going to do this thing in the case of your bread. So what happens, guys? So what are you going to do when you are going to prepare the bread? Okay, like the dough and all. Let's try to see what exactly is happening out over there, guys, at that time. Okay, so first of all, how do we prepare chapati, guys? Okay, it's exactly the same process how you're going to prepare bread. Okay, so what are you going to take? You are going to take some flour. Okay, so you have some flour. Okay, to that you are going to add little bit of your salt to taste. Plus, you are going to guys, is the screen blur or is it okay? No, okay, 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 okay. See, guys, once again, when you make chapati, how do you make chapati? It's the exact same process of making your bread as well. So, if you have seen, we are going to take some flour. In that, we are going to add some water. We are going to add some salt. Okay. And then, what are we going to do? We are going to add a little bit of oil. And then, we are going to take that and we are going to knead it into a dough. That is what is going to happen in chapati making. Same way we are going to do this thing out over here. So we take some flour, we add a little bit of salt, okay, for taste. We are going to add water. And now what happens is, guys, we are going to start kneading. Okay, kneading meaning you are going to hold it in both your arms and you're going to squeeze and squeeze and squeeze till then, I mean, till you get that sticky nature. Now, guys, while this thing is happening, okay, so see, guys, what are we talking? We are talking about bread preparation, okay. Now, what are we going to do is, guys, on other side, we are going to take yeast. Now, those who are into baking and all, guys, you know that we get this baker's yeast. Okay, these are like dried powder, and what do we do is like this baker's yeast to activate it. We are going to add it into this thing. We are going to add it into your little amounts of your sugar solution. Okay, so you're going to take little bit of your sugar, add water to it, and then add baker's yeast, and you are going to just mix it completely to make it a paste like thing. Now you're going to take this yeast and you are going to add it out over here while you are kneading it. Okay. Now what happens, guys? When you are going to knead this thing continuously, thoroughly, we are going to get the dough. Okay, fine. Chalo. So we have got the dough. So guys, now let's say your dough is like this. Now at this moment there is nothing. Okay, you have just taken the dough and you have put this thing in a utensil. Can I zoom out or you want to zoom in? Yes, see, you're absolutely right. That is the next thing that we're going to talk about. Arya, you want zoom in or zoom out? This is zoom in and this is zoom out. Okay. Okay. So now, guys, what have we done? We have taken our dough, okay, and we have kept it into a utensil, just like this. Okay. And now we are not going to immediately use it, guys. Okay. Inside this, what are we going to see that, oh, you are going to have this yeast. Okay. So this is all active out over there. Okay. So yeast is all going to be present. Okay, th- there's a little problem. Okay, so now what is going to happen is, see, the yeast is already present. Now what are we going to do? We take this dough and we rest it for a while. Okay, for a couple of hours. Now during this couple of hours, what is going to happen? See, this entire dough which was there, guys, you are going to see that now this dough is going to increase in size. How? Let's try to see. So after some time, when we come back, guys, the same dough, you are going to see that, oh my god, this has like completely risen almost three times its original size. And plus, what are you going to see is that this dough, it becomes like, really fluffy, like this. Now, how does this thing happen, guys? See, the reason for this is, see, when we talked about your yeast, okay, the yeast was now activated. Okay, what is going to happen? The yeast is going to eat away all the sugar. But where is the sugar present in this thing? See, guys, the moment we had used flour out over here, this flour, it contained maltose. Okay, because flour is going to have malt is nothing but malt sugar, that is maltose is going to be used immediately. It is going to be broken down into glucose, and now the yeast starts utilizing glucose. But as it utilizes glucose, what is it going to do? It undergoes anaerobic respiration. Guys, what is going to happen in this anaerobic respiration is C6H12O6, directly in the absence of your oxygen, it is now going to be converted into 2C2H5OH plus 2CO2, 2C2H5OH plus 2CO2. Guys, what is this? So you are going to see carbon dioxide being formed. Where is that carbon dioxide? That carbon dioxide is out over here. So it produces a large amount of carbon dioxide, and this carbon dioxide, it is going to get trapped. It gets trapped into a protein called as gluten. Okay, and because of this, now what are you going to see, guys? You will see that because of this, therefore, the size of the dough increases, and increases to how much, guys? It increases almost three times. It increases almost three times. And what is this called? This process is called as leavening. Increase in the size of the dough. Okay, but now, guys, see, what is the other component that you have produced? This is going to be alcohol. Arey baap re, ethanol. Oh. So that means your bread must be having ethanol in it, right? So my question to you guys is, guys, after you have bread, okay, after you have bread, are you high? Like suddenly you are like, oh my god, oh, ये बहुत अच्छा लग रहा है. No. So where does this alcohol go? What happens to this alcohol? Because whenever you have bread, I don't think so. Like, you are going to sway. The reason for this, can anybody give me the reason? Anyone? Share. 1 minute. By mistake, I closed that entire this thing. Sorry, 1 minute. Absolutely. Ashwin has given me the right answer. Guys, remember, our our bread is not yet prepared. Guys, this is just the dough which has increased in size. This is just leavening. But guys, after the dough has increased in size, what is the next step? We are not going to eat it directly. We are going to put this thing in oven, guys. And now the moment you put this thing in oven, what is going to happen? Whatever little alcohol, whatever that was there, that is going to evaporate. Okay, now that is going to evaporate. But guys, at the same time, you're going to see that carbon dioxide that is going to be there, this is going to expand, and immediately this is going to leave it. Okay, and leaves the dough. But now, guys, what happens is the moment it expands and it leaves the dough, what is it going to do? It is going to make the dough porous. It makes the dough porous. And whenever you have bread, guys, inside that bread, you are going to see that there are these small holes. So let's say we are talking about Wibs bread, or you're talking about modern bread. So you have these tiny pores structures, guys. These pores are nothing but the the carbon dioxide gas has passed through this thing, and it has made these small holes out of there. This is what we need to know with regards to your bread preparation. Everybody all right with this thing? Got this thing, guys? Sure. Now, from here, what are they going to ask you, guys? They will always ask you, what is gluten? Okay, gluten is nothing but this is going to be a protein which traps the carbon dioxide, okay, and prevents it from escaping. And therefore, it is going to help in the leavening process. This is very important. Okay, everybody all right with this thing? Sure, no problem. Got this part properly, guys? Okay, but now, guys, the next thing is, now, is this the only product that we are going to make it, guys? Is this the only thing? The answer is no. Okay, so where does the alcohol go? It is going to evaporate. It is just going to evaporate into your oven, and then it is going to be just puffed off into the atmosphere. Meaning, then the oven is going to have an exhaust, maybe, or something. So from there, it is going to go out. Okay, and remember, the amount of alcohol produced in this is not like really huge. कि you will think कि अरे चलो इसको हम लोग ना इधर fermentation करते हैं इसका, and then when we put it into the oven, whatever the heat that is or the gases that are coming out of the oven, we will bring about distillation and बन गया दारू. No, you cannot do that, because the amount of alcohol in this is very low, very little. Okay. Yes, Devang, right. Okay. Sure. So, guys, this is the first thing. This is going to be your bread preparation. I hope everybody has understood how this bread is prepared, guys. Okay. So, once again, just have a look if everybody is okay. Then we'll move ahead with the next part. Okay. Sure. So, guys, let's try to see the next thing. Okay. The next one is like, your idli dosa. Okay. Idli dosa mixture. How you make that stuff. Let's try to see. Okay, so guys, the next one we are going to have your idli dosa dhokla. Okay, all that thing. Okay, so dhokla, dosa, idli batter. Okay, once again, we are going to use exactly the same kind of things out over here. Okay, so guys, generally, what are we going to do? So, especially if you're talking about dhokla. Okay, so let's say we are talking about dhokla. For dosa and idli, you're going to use different dals, urad dal, tandoor, ye, wo, so, and then you're going to mix it. You're going to keep it overnight. Okay. Okay, so now guys, in dhokla, let's imagine what are we going to do? We are going to take this gram flour. Okay, gram flour is what? Nothing but it is besan. Okay, so we have taken gram flour, that is besan. And now to that besan, guys, what are we going to add? We are going to add a little bit of buttermilk. Okay, now why are we adding buttermilk, guys? Because buttermilk is going to contain lactobacilli. They contain lactobacilli. Now, guys, some of them, okay, and I'm sure if some of you are into like cooking and all, what you do is you're going to add eno to it, okay, or you will add like baking soda, okay, eno, baking soda, or for others, what you will do is you will take like actual soda. Okay, soda water basically, and you will put it into that, and you will mix it with soda, so it becomes like, fluffy and airy. Okay, but then let's say we are talking about the normal stuff out over here. So we have taken gram and we have taken some buttermilk and we are going to mix it. Okay, now you can have this thing for uh, cakes, but cakes is like more like the dough kind of thing. Yes. Okay, and the result remains exactly the same, guys. Okay, the result remains exactly the same. Now what are you going to do? See, immediately after you make this thing, guys, you're not going to this thing. Okay, you're not going to utilize it. So now you are going to allow it to ferment. You allow it to ferment, and the moment you allow it to ferment, guys, after some time you can now make this entire batter. So initially, like, the batter would be this much. Okay, so once again you are going to see exactly the same thing. The batter is not so airy, the batter is not so thin, and batter is not like fluffy kind of thing. You can see it actually. But now, after keeping it at room temperature, maybe overnight, what are you going to see? Now, once again, you are going to see that, oh, the batter is has like risen up. Okay. Plus, you will also see that, if you taste this batter, it will be khatta. It will be khatta. Why? Because it will be sour because of your lactobacilli. The lactobacilli, they are going to increase in the number, and plus this is going to produce as acids and all, and plus there is going to be this little bit of carbon dioxide. Okay, little bit, or not much kind of thing, and this is going to rise up. This is what is going to happen. Then what are we going to do? We take this batter, and now let's say we are making dhokla. So what are you going to do? You are just going to steam it, and the moment you steam it, guys, we have got a nice dhokla, nice and fluffy dhokla. Okay, everybody all right with this thing, guys? So this is nothing but your dhokla preparation. So, guys, which are the organisms that we are going to use for this kind of dhokla, or maybe this fermented products? So generally the organisms that we use, guys, we are going to have lactobacilli. Okay, so lactobacillus. This is the most common. Okay, this is found in your milk. These batter also contain alcohol before steaming it. No, 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 no. Because see, who are we using? We are using lactobacilli. Lactobacilli produces lactic acid. It does not produce your this thing, your alcohol. See, in this thing, this is going to ferment, but the fermentation reaction, how it is going to be? C6H12O6, in the absence of your oxygen. Now this is going to give rise to your 2 lactic acid. It is just going to be 2 lactic acid plus your 2 ATP. So here you're also not going to see that, the carbon dioxide and all that is going to be huge in amount. This is what is really going to happen. Okay, but here what are you going to see? That you're going to feel that, it is like, nice, really nice and sour. Okay, so we are going to make use of lactobacillus. We are going to make use of your bacillus. We are going to make use of candida. Now, guys, candida is a fungus. Okay, candida is a fungus like your yeast, from the yeast family, and then you are also going to make use of yeast. But guys, now what is the name of yeast, guys? The name of yeast, basically, this is Saccharomyces cerevisiae. Saccharomyces cerevisiae. Okay, guys, always remember this, because we are going to require this name again and again, again and again, again and again in the next part of the chapter. So this is going to be Saccharomyces cerevisiae. Okay, so first, guys, this is in your household industry, that is your household microbiology, or in your kitchens. Okay, so very, very plain and simple, guys. For your dosa, also, what you're going to do, like, if there are like South Indians, or if you are into dosa making and all, you're going to take some ratio of your dal. Okay, well, this is what I've seen. Like, you have some urad dal and some other dal. Okay, you have rice. You are going to mix that thing up. You're going to grind it, add a little bit of your buttermilk, or you just leave it that way overnight. You just leave it overnight that way, and next day when you see the entire batter becomes like nice fluffy, it becomes soft and airy. Okay, so once again the process remains exactly the same thing, guys. Beer, wine are also examples of fermentation only. We have this thing in detail, so we will be seeing it in the next class though. Okay, beer, wine, alcohol, that is your whiskey. We will be seeing it in the next class. Okay, but now guys, chalo, this is fine. This is going to be your dokla and somewhat like your bread dough. Yeah. Okay, but now guys, just like this, we are also going to make use of your microorganisms in your dairy industry. Okay, so in dairy industry for what? Okay, let's try to figure this thing. Absolutely. So first of all, guys, the first thing that we are going to use it is, see, in the dairy industry, which cultures are we generally going to use, guys? We are going to use especially your lactic acid bacteria. Okay, these are the most important one. Lactic acid bacteria, we also call this thing as LAB or lab. Okay, lactic acid bacteria, they're known as lab. And in lactic acid bacteria, the most famous one is your lactobacillus. Okay, this is generally used to milk. Okay, now generally used to prepare your milk or something. Okay, so what happens is we are going to take this thing and we are going to add it to your milk, and this is going to give that milk a different texture and different shape, structure kind of thing. Okay, now what happens, guys, the moment lactobacillus is added into the milk, the lactobacillus, it starts producing lactic acid. Okay, now understand this thing, whatever we are talking about. Okay, guys, just understand this. Okay, the lactobacillus, it produces lactic acid. Okay, but now, guys, if you are producing lactic acid, now my question to you guys is, see, guys, have you ever seen that whenever you take some lemon and you squeeze it into milk, what happens? The milk starts separating, the curd starts separating out. Okay, and what we get is paneer after that. Exactly the same thing, guys. As the amount of your lactic acid increases, immediately you are going to see, no, this is going to be in your dairy industry. This is the second thing. This is second part. We are into dairy now. This is dairy. Okay, now what is going to happen, guys? Immediately you are going to see that whatever proteins that are there, the protein starts coagulating. Okay, so you will see that the casein that is there, especially the milk protein. Okay, so the casein, it starts separating out. Okay, or it starts coagulating. And now, as the protein coagulates, now what are you going to get, guys? From here, you are going to get curd. This is nothing but our normal dahi. Now, guys, this dahi can be converted into, okay, this can be converted into your paneer. Okay, paneer is what, guys? It is going to be your cottage cheese. Okay, so this can be converted easily into cottage cheese by further processing, or you can convert this into your cheese as well by further processing. So this is what we can do this. Okay, so guys, for this entire curd formation, okay, so whatever we were talking about, this is curd formation, right? So for this curd formation, guys, what are we generally going to add? Okay, so the organisms used for this entire curd formation, we are going to have, as I said, we are going to have lactobacillus. But in lactobacillus, guys, the most famous one, we have lactobacillus acidophilus. Lactobacillus acidophilus. Okay, so this name you'd remember. Okay, now there are many bacteria. Okay, like we have Lactobacillus bulgaricus, Lactobacillus casei, Lactobacillus your sake. Okay, so those are all the other bacteria. We can use it, okay, in this thing. Okay, but then the most famous is Lactobacillus acidophilus, and we have the other one, Bifidobacterium. Okay, so these are the two most important ones. So guys, this is what we have in curd. Okay, so this was our curd. But now the same thing, guys, we can produce yogurt also. Okay, but now when do we generally call it as yogurt? Now see, generally yogurt and curd, they are almost on the same lines. Okay, a little difference, not much. But when we have to prepare your yogurt, guys, once again it is going to be prepared by curdling of milk only, like how we did it in the upper case. So this is also curdling of milk. But now, what are we going to use out over here? The organisms that we are going to use is different. We are going to use Streptococcus thermophilus. Right? Now, why is the name thermophilus? Guys, because the Streptococcus, this can withstand extremely high amounts of temperature. And how does this thing appear, guys? This Streptococcus, basically, this will appear to be in long chains like this, and they can withstand extreme temperatures. That is why Streptococcus thermophilus. Yes, you get them everywhere. These are also like very very common. Okay, so along with this, guys, add the other one plus. Okay, another one that is going to be important. That is going to be your Lactobacillus bulgaricus. Okay, now you will say, sir, are all these names important? The answer is yes. Okay, so these names are important because in your exams they will ask you this thing only. Okay, they will ask you this, that okay, who is produced by what method or which organism. So you need to remember this. Okay, but then after this, guys, we are also going to make use of the same curd for making your buttermilk. Okay, so how are you going to make buttermilk, guys? You are going to take your dahi, that is, you're going to take your curd, and plus to that, you're going to add water. And now, what are you going to do? You are going to churn this thing. Okay, you start churning and churning and churning and churning. Okay, and then after churning for a little while, you are going to see that the liquid, it is going to separate out. Okay, so the liquid, it separates out, and this is nothing but your buttermilk. This is going to be buttermilk, or we also call this thing as chas. Okay, but now the next one, guys, the next one, the last thing out over here, we are going to have cheese production. Okay, so let's try to see how is this cheese going to be, what are the different kinds of cheese. So guys, the first process remains the same, like how we made the curd out over here, it remains the same. Okay, I will tell you a little quick way in which how we are going to produce cheese. Okay, so guys, first thing, what are we going to do? Let's say, okay, now I'm showing you in a diagrammatic form. You can write it down in your, a paragraph form also. So that is not an issue. So guys, we are going to take some milk out over here like this. Okay, and to this milk, we are going to add either from outside or naturally present also. A streptococcus and lactobacillus is already present in yogurt, right? Yes, it is naturally present. That is why I'm saying it is already present. Okay, so there is no need to put extra thing. But then what happens, see, when you want some premium product or when you want some consistency in your products, you cannot just rely on like अरे उसमें हो सकता है, उसमें होगा. And now that cannot be done. So you have to maintain the quality, and for that you will always select some specific kind of your bacteria only, and that bacteria is maintained generations and generations and generations, so that the consistency and the flavor profile of the product remains the same, it doesn't change. Okay, so guys, we have taken milk out over here, and now to this milk we are going to add your lactic acid bacteria. Okay, it can be your lactic acid bacteria, that is like your Lactobacillus bulgaricus. It can be your Lactobacillus casei. It can be your Lactobacillus sakei. Okay, so it can be anything. Okay, so okay, chalo. So we have added this lactic acid bacteria. Now, in the next moment, guys, what is going to happen as we allow this thing to ferment, guys? Okay, you are going to see that, oh, suddenly all the proteins, they are now going to coagulate. Oh, okay, so now let's imagine the proteins have coagulated and the liquid has separated out, because at this moment you will see this liquid coming out. So you will have the solid part, okay, which we like to have, and then there is this liquid part which is going to be present. Now what is this liquid part called, guys? This liquid part is called as whey. Okay, in fact, if anybody of you is into your bodybuilding, okay, you may have heard whey protein, right? This is nothing but that whey. Okay, so we have got our curd. Now what? Now what are we going to do is, guys, we are going to remove this entire whey. Okay, so in the next case, we are going to just have the curd. Okay, so you have removed the whey. Okay, and we have a situation something like this. So the whey has been removed. Okay, now the way, after you remove the whey, okay, we have got, this is nothing but a normal curd. You can just have it as like this only. But no, are we going to stop out over here? No. Now what are we going to do, guys? We are going to take a, a plunger kind of a thing. Okay, now I'm there are many ways. Okay, we can do this thing. I'm just showing you one way of doing it. Okay, now this curd, guys, this is still going to be little moist. It is still going to be like, very like, semi solid kind of a thing. Now what we do is, guys, we are going to take a plunger, a wooden plunger kind of a thing. And now what are we going to do? We start squeezing this thing. Okay, now this lower part, it has like small holes inside. Okay, so it has like, really minute holes. You can just imagine it to be like, a semi permeable membrane kind of thing. So now what happens is you start applying pressure from the top. So you apply pressure and pressure and pressure. And because of this, now what happens, guys, you are going to see that all that extra wave, whatever that was there, all of that is going to be removed off. Okay, and now as you have removed of that entire way, guys, now what are you going to get? Now after doing all this thing, guys, you are going to get a little flat structure. Okay, because you are like pressed it from the top. But now, guys, what is this flat structure that we have got? Yes, we have got paneer. Okay, this is nothing but paneer. After which, we have like, from the curd, when you remove the entire moisture, we get paneer, or we call this thing as your cottage cheese. Okay. But now, once again, guys, our job is not yet done. We have to convert this into your cheese. So, what we do is, now we are going to take this paneer. But still, this paneer, guys, if you look at it, it is still moist. It is still like, it is still fluffy kind of thing. But if you look at cheese, is it going to be moist and it is going to be like, easily, you can break it with your fingers or something? No. So there is a lot of moisture out over here. So what we do is we take this paneer, and now we are going to have these huge vessels with brine solution. Okay, these are going to be huge vessels with brine solution, salt solution. Okay, so we have excessive amounts of your salt solution, highly concentrated, meaning saturated solution of your salt. Now what we do, guys, we are going to take this paneer and we are going to drop it out over here. Now, guys, what is going to happen? See, whatever water that is present inside this paneer, all that water is going to zip up. Okay, you are going to see that all this water, zopuk, it is going to be removed out like this. Okay. And the other thing that is going to happen, guys, you are going to see that because you're putting it in salt, whatever, like, germs and bacteria and all that may be present onto the outer surfaces and all, immediately they are going to get killed. Okay. Now, guys, okay, 1 minute. 8 to 10 months for cheddar, 18 months, then we get parmesan. Yes, yes, yes. There are different timelines, different flavors. The older the cheese, the better it is. It is that way. Okay. There are different kinds of cheese. Like it depend on their fermentation. Yes, 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 yes. Absolutely. Okay. So now I'm showing you like only one method, that is traditional method. These days we have like different methods out over there. Okay. Now, guys, once all this water has been removed, then yes. Okay, once all this water has been removed, guys, now we are going to get one slab like this. But now at this moment, now, guys, there is no flavor as such. There is no flavor. So what are we going to do is we are going to take this slab of cheese. Okay, this is like partially prepared cheese. And now we are going to bring about a ripening. Okay, so the next process is we are going to ripen it, meaning we are going to put it on a shelf and we are going to let it ripen. So let's say this is our table kind of a thing. You are going to just let it be there, maybe for 1 year, maybe for 2 months, 3 months, what, how long you want it to be. So the next step is ripening. And during this ripening, guys, what are you going to do? You are going to sprinkle fungus on top, like your penicillium. And that penicillium or the fungus, guys, as you keep it for ripening for years and years, that is the one which is going to give it that nice exquisite taste. Okay, so you are going to add penicillium or any other fungus, okay, the one that you have been using for. Okay. Okay. So ripening. And now after few months, okay, after a few months, that depends once again, like, how many months you are waiting. Okay, so after few months, now our cheese is ready. Then you are going to have this nice cheese. So what are we going to do is we are going to remove the outer covering, because the outer covering was in touch with your penicillium and all, right? So that is going to be removed off. And then we are going to have this cheese, and this cheese is now ready for distribution. Yes. Sir, those holes in the cheese are also caused by fungus? Yes, I'm going to talk about that as well. It is basically, your bacteria. Okay, there's this bacteria. But then if you have seen a Rockford cheese, again, I will talk about this, then you will get it. You will get it. Okay, but first, just see if everybody is okay with this cheese production, guys. Okay, when you are going to go for ripening, guys, you have to maintain the temperature, you have to maintain the conditions, the moisture, everything. Okay. Just a general question. Now these days there is a debate of people shifting to vegan milk, even though cow milk having so many benefits. Is it just for animal protection, or like vegan is better? Now, frankly speaking, it is my personal opinion, it may not be in line with what you think. It is all, shoshay, I feel. Okay. What suddenly people have started to do is like, they have started like, no, no, aisa nahi hai, waisa nahi hai. I want to bring about a change. I don't want to harm, any animals. I don't want to. But no, it is not that way. See, what happens is in this world, see, we have like, hierarchy. Okay. So one person is going to eat the other person is going to eat the next person. So it is that way. And when you are having milk from the cows, okay, we are not really harming them as such. Okay. So cow milk has definitely huge amounts of benefits. Vegan milk, okay, so some people may not know what is vegan milk. Vegan milk means what? It is almond milk, soy milk. Okay, those are nothing but, alternatives, like plant-based milk products. But then it is not exactly of the same type, guys. Sir, but it's life cycles. It's a life cycle. So by becoming a vegan, we are disturbing cycle. Yes, absolutely. You are going to be disturbing the entire cycle. At the end of the day, we have more canines. Yes. Yes, it gets more costly also, as well as the yes, vegan milk. Yes, vegan milk is more costly because you're using, see, for from almonds, if you're going to remove milk out, definitely it is going to be more expensive. It is not going to be cheap, right? So there is a huge difference. Okay, and yes, then it is just PETA wants its profit. Yes, absolutely. See, it is always that, we are born. See, I am not against, like, the vegetarians and all. Okay, you eat plants, I am happy with it. Okay, but those who are non-vegetarians, they should continue their lifestyle. Aisa nahi hai. Okay. Yes. Okay, anyways, so guys, this is how your cheese is going to be made. Yes. Okay. Sure. But now, guys, let's try to see, how are these cheeses, which of them are, like, who is going to have, like, which bacteria or which thing inside of it. Okay, which fungus. So we have some famous cheeses, guys. Okay, so we have, okay. So you must have always seen that this, we have Rockford cheese. Okay, so this Rockford cheese now very very famous. It is like, the blue green kind of cheese, like, inside it has nice bluish holes. Okay, so that is your Rockford cheese. Okay, this Rockford cheese, this is prepared with the help of your penicillium, Penicillium rockforti. Okay, yes, yes, yes. I'm just finishing this thing off. Okay, so this is going to make use of Penicillium rockforti. Okay, then we have the next one, that is your Camembert cheese. Okay, somewhere on the same lines, Camembert cheese. This is going to use the same kind of thing, but Penicillium, but now it is going to be Camembert. Penicillium Camembert. Then, guys, you must have seen, okay, your uh, Tom and Jerry. Okay, in Tom and Jerry, Jerry is always in moving through that cheese, right into the holes into the cheese. What is that? That is nothing but it is your Swiss cheese. Okay, that is Swiss cheese. Okay, so I will just mention out over here that it has some big holes inside. Now, how do we get these big holes, guys? This is going to be because of carbon dioxide production, and excessive carbon dioxide production by whom? It is going to be by your Propionibacterium shermani. Okay, so this is going to be by Propionibacterium shermani. Okay, so this is what we need to know with regards to the different types of cheese. Okay, everybody all right with this thing? Okay, sure. So guys, with this we are done with the first part out over here. Okay, so once again, when we come back, we'll continue with the next thing, that is how we are going to make use of mushrooms, how we are going to make use of single cell proteins, how we are going to make beer, wine, alcohol out over there. So that is all in the next class, guys. Okay, sure. So once again, we'll stop over here for the day, and I'll see you in the next class. Till then, take care. Bye bye. Yes. Propionibacterium shermani. Yes, yes, yes. Bye bye. Yes, 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 yes.